Alright, so there's going to be a couple ideas we talk about in this video. To start out, we're going to open up with polymerization. Should be a little bit familiar at this point. We've talked about it with nucleic acids. And it's just the same idea. We're going to take our monomer, which in this case is an amino acid, and we're going to make a polymer out of it by attaching them together covalently. And nucleic acids were actually a little trickier with this because it had the sugar phosphate backbone and the nucleic acids actually branched off of that. In this case, it's just amino acids. It's just a train made out of train cars. Just boom. They all stack together. They all attach the same way. So once you know how to put 2 together, you know how to put 200 together. You know how to put 200,000 together. It's going to be the same way. And so it's going to be, guess which type of chemical reaction it's going to be. That's right, it's a condensation reaction. Someone probably said hydrolysis. It's okay. It always happens when I teach the class live, so it's not a big deal. So condensation reaction is going to link this together. It's always condensation reactions. Any of these polymerizations with biomolecules, condensation reactions happening over and over and over again. And so if I keep this simple, I'm not going to draw anything with crazy side chains. What we have, so I'm going to do a glycine and an alanine. So I'm going to draw my glycine. So guess what? That doesn't change. I don't know if I've made that clear yet. Okay, so I've got a glycine. And my other reactant. Oh, actually, I'm going to get a little more specific here. So that's a single bond, double bonded O. Okay. And then I've got an N with 3 H's here and a positive charge. So to be clear, that's a single bond. Sorry, I forgot that I need to draw it a little more detailed just to help illustrate what's going on. This is my alanine. So I have 2 different ones here. And remember, we can do condensation reaction here. So we can get this oxygen and 2 of these hydrogens. Doesn't actually matter which 2 as long as they come off the nitrogen. And that will leave and make a water, H2O. And draw my reaction arrow down here because I'm out of space over here. And what I get is and a water. So this is called a dipeptide because there's 2. Polypeptide is the generic term, or polypeptide chain. It's just a chain of amino acids linked via condensation reactions. Alright, and so if I'm looking at this, how do I know, let's say I have a longer chain, how do I know how many amino acids are in it? Well, first I just identify where the linkages are. I don't worry about the side chains. They're going to be sticking out wherever, like maybe it loops around, it does all sorts of stuff on the main chain. And then this is my trick. When I want to know how many amino acids are in a chain, I just count the nitrogens. There's only 1 nitrogen for every amino acid in this chain. There's 1, 2. Now, that's why you don't want to follow side chains, right? Because you could have a basic side chain that has nitrogens in it. So you don't want to get lost there. You want to be able to just continually go through and just follow along and just count nitrogens. Could be 20 or 9 or 13. In this case, it's 2. And this is a process that we could repeat over and over again. Look, we could add on to this end. We could add on to this end. We don't add to the middle. So we just keep stacking in amino acids. And the process for doing this, you'll learn about in your biology course, or maybe you already have. And so that is translation. So it's heavily controlled environment. RNA is red. There's 3 bases. And that combination of 3 bases tells describes what amino acid should go in that spot. and then the amino acid is brought in, if I remember right, with tRNA. It's all connected together. So it's not just like this is happening freely in the cell. This is being heavily controlled. So there is like a factory that is pushing these amino acids together and making these bonds happen. So, and then in the next section we'll talk about what happens when this thing's fully built out. Then what happens to make a protein, because you're not there yet. There's a lot more work to do still. Okay, so that's the first idea. So polymerization is just a condensation reaction. I'll write this out. Between the carboxyl and amino groups of two amino acids. And then you can repeat it over and over again. So we can just keep adding on. This is available for a condensation reaction. This is available for a condensation reaction. So you can build it out. Now you're only going to go one way. I forget which one it is, a translation. Like I said, I worked hard to forget this stuff, but essentially it just gets built in one direction in the cell, in reality. Alright, so that's polymerization. I talked about, how do you count? Well, you just boom, boom, 2 nitrogens in my chain, 2 amino acids. Alright, so next let's talk about phosphorylation. So this is an important idea in amino acids, and it's really important once they're proteins, because phosphorylation is a technique that cells can use to turn off or turn on proteins. So it's really nice to not just build them as you need them and break them down when you don't, but maybe to have a protein sitting there idling, and then you phosphorylate it and it turns on, and then you dephosphorylate it and it turns off. Whatever that means, because I haven't defined the word yet. So there is a clue in the name. And so phosphorylation just means attaching a phosphate, that's where the phos comes from, or the phosph comes from, to a side chain. Oops. Via, guess what type of reaction? Condensation reaction. So if you don't have a side chain that can perform a condensation reaction, then you can't phosphorylate it, right? So there's a little note we'll make here. Side chain must be able to, we'll say, participate. So it has to have like an OH or an NH group on it, or a carboxyl group, which has an OH group on it. So it has to have those to be able to participate in a condensation reaction. If it can't participate in a condensation reaction, it cannot be phosphorylated. Oops, condensation reaction. Okay. So here's what we're talking about. For example, let's bring our old friend serine back into the picture here. Where'd serine go? Make sure it draw it right. There it is. So we go draw it in the Zwitter ion form, and I'm going to get a little more specific here. So it has the OH group. I'm going to draw the OH group coming off of there. This is serine. And so we're just looking at the side chain. Because you're like, oh, well, I know it can perform condensation reactions because of these points. Well, yeah. But we're talking about with the side chain. So can we add a phosphorus to this via a condensation reaction? And the answer is yes, because there's an OH group. And so what we can do, we've got our phosphorus, our old friend from the nucleic acid stuff. Well, that will make a water. We got an H and an O, that'll make a water. And so what we get is, and generally this doesn't happen, by the way, with just a free-floating amino acid. I just want to show it with that. But generally it happens in the protein when it's already fully formed. But, you know, can an amino acid perform a phosphorylation? Well, basically what we're asking is, can the side chain perform a condensation reaction? The answer is yes, the side chain can perform a condensation reaction, and the answer is yeah. It can be phosphorylated. So, that's what it comes down to. But this is what it looks like. And this can be a very important mechanism for switching on or off proteins. Sometimes you phosphorylate a protein, turn it off. Sometimes you phosphorylate a protein to turn it on. It just depends on the situation. And we're not going to get into that much detail on it. But it is a really neat mechanism because we tend to have phosphorus just floating around. It's a good way to use it. Alright, and let's see. Is there anything I want to add to this before I go on to the next topic, just to be safe? So, it's a little bit tricky. We'll talk more about phosphorylation too when we talk about ATP and ADP, so the ATP cycle, the cash money of energy in the cell. So it's not totally gone after this, but so just be aware. You're just looking for OHs, NHs on the side chain. If they're there, it can be phosphorylated. If not, it can't. Alright, so the last thing we're going to talk about here, the last thing for this section, kind of last detail of amino acids, are, oops, always helps to have, you know, a pen uncapped, are what we call disulfide bonds, or sometimes they're called bridges. Alright, and so we want to bring back our old friend cysteine. But before we do, so what we have are, these are bonds that form between two cysteine amino acids, because both of them have to have SH groups. They both need thio groups. And so it's actually a covalent bond. So it's a very strong way of holding two amino acids that aren't in the chain next to each other. They could be thousands of amino acids apart, but you can connect them with a disulfide bond. And a covalent bond is stronger than any attractive force. So, what you get is, so let me check my notes here, make sure I don't mess this up, because I got to draw one upside down. But you're going to have two cysteines here, and we're not going to worry about the mechanism. I'm just going to show you what it looks like. And so it's called a disulfide because it's actually going to use both sulfur. So it's going to be sulfur bonded to sulfur. And so we could have something like this. So I'll draw my backbone. And then we've got a CH2, right? 'Cause it's going to be a cysteine. And it goes to a sulfur, which goes to a sulfur, which goes to a CH2. And so, whoops, I tried not to mess it up and I still, there we go. There's CH2 in the middle there. Okay, so this is the disulfide bridge. And so if you're asked the question, like, can this form a disulfide bond or a disulfide bridge? Just go, well, does it have an SH, right? Is it cysteine? So remember, cysteine is man, I really painted myself in a corner here. NH3, I can do that. I'm a professional. I'm allowed to. It's cysteine that does this. Yeah, the question is it cysteine? But really, you don't have to memorize the structure of cysteine. Just go Does it have that? Does it have an SH group on it? If it has an SH group on it, it can form a disulfide bond or disulfide bridge. That's two names for the same thing, to be clear. And this is a covalent bond. I drew a line there for a reason. These are covalently bonded to each other. So, this is kind of the last one, and these are just sort of, just kind of thrown in. So the main one is like polymerization, and then there's some extra tricks, and these are going to come up more. I'll talk about disulfide bonds more in the next section as well as the after effects of once you've polymerized. Phosphorylation, just can an amino acid phosphorylate or not? We're not going to build on that more for now. It'll be really important for ATP and ADP, but that's not for a little while yet. That won't be till well after biomolecules. So that'll do it for this section. So it feels like a lot of details, but really it's just really narrowing it down to what am I looking for? I'm just looking for that. Okay? If it does that, if it has that, it can form a disulfide bond, but it has to be two. It doesn't just form a disulfide bond with another amino acid. It has to be another amino acid that has that. And it turns out there's really only one. Phosphorylation. Well, I'm just looking for an OH or an NH because phosphate will be able to do a condensation reaction with those. And then polymerization. All the amino acids can undergo polymerization. That's what this backbone enables us to do. And then we get something like this, and how do we know how many amino acids are? We just count nitrogens. So that's what all this comes down to. We will be building more on these ideas in the next section. Amino acids and proteins are essentially the same thing. We just broke it up because you can to talk about them, I guess. I don't know. When I teach in class, it's just like it's amino acid day. That's the same as protein day. But a little bit different approach here. So I've had to shuffle things a little bit, but I think we're getting through it okay. But as always, if you have questions, use the help boards. Email me, come to my office hour. Whatever I can do to help you out, just let me know.